Boom. So what about sympatric speciation happening without chromosome funnies? Is it possible to have a population that just kind of divides into two without being split by a barrier? The stock answer used to be no. That was beginning to change as I was coming up in grad school. And we now recognize that it is possible for speciation to happen without geographic isolation. This was one of the first examples to be well studied. You're looking at an apple with a little maggot in it. That's the larva of the apple maggot fly, Ragoletus pulmonella. By the way, that's an adult apple maggot fly. Now, the way their life cycle goes is the adults lay their eggs in fruit. The larvae feed on the fruit. This is why this is an agricultural pest, because having larvae in the fruit means the fruit's not sellable, right? Nobody wants to buy, apples with maggots in it. The fruit eventually falls to the ground, and mature larvae leave the fruit, burrow into the soil, pupate, right? Become the similar stage to the cocoon that you'd see in butterflies and moths. And they'll hang out in the soil as pupae, and they'll spend the winter in the soil. And then they'll spend the winter, they'll spend most of the spring, and then the adults emerge in late spring or summer. The adults will fly around and do their little fly thing, mate, lay their eggs on fruit, and round goes the great circle of life that moves us all. Adults can lay eggs on a variety of fruits, but we'll focus on two main races of Ragoletus pulmonella that have been established in the north central United States. The apple maggot fly and the hawthorn maggot fly. Apples, hawthorn may not be as familiar to you. This is genus Crataegus. It's a close relative of apples. The fruits can be eaten. If you've encountered them, it's likeliest, okay, there's a hawthorn species we have in the South called the mayhaw, and the fruit is used to make jelly. It's the kind of thing your meemaw, your grandmother, might have known about, mayhaw jelly. Anyway, so Ragoletus pulmonella, though still one species, has these two subvarieties, or they're sometimes called races. They're just varieties that primarily feed on only one species of fruit. There are apple maggot flies and there are hawthorn maggot flies. And they've been studied pretty intensively up in northern Michigan. And here's what's unique about them. Apple Ragoletus adults emerge mostly in mid-July. They'll start emerging by about mid-June, and they'll reach their peak round about here. That's roughly third week of July, between about the 15th and the 25th. That's mostly when most of them will emerge. And then there's a few stragglers that will emerge after that, with maybe the very last coming out at the beginning of September. Here's the thing, that's 6 weeks before the maturation of the apples. This is when the apples are maturing. A few apples will come ripe as early as late July. Apples will hit their peak up there around about the beginning of September. And the very last of them might be ripening in round about the beginning of October. But notice you have that 6-week gap between the maggot flies emerging and the apples coming ripe. Okay, so because the adults take about 6 weeks to sexually mature before laying their eggs on the apples. So the adult apple flies come out of the ground about 6 weeks before the apples are ready. Okay, the hawthorn flies don't start emerging until August. They're at their peak emergence at about the middle of September, and the last of them don't emerge until about the middle of October. That's also about 6 weeks ahead of the hawthorns, which start ripening in early September, hit their peak in mid October, and aren't done ripening until we hit the about middle November. So notice something. What's separating the apple maggot flies and the hawthorn maggot flies is not a barrier in space. This is all happening in the same parts of northern Michigan. It's a barrier in time. The apple maggot flies are mostly all emerged before the hawthorn flies have even started. The apple maggot flies are laying their eggs at their peak right about the time the hawthorn race is starting to come out. There's only a very narrow window where apple maggot flies and hawthorn maggot flies would encounter each other in the first place. And, apple maggot flies, very few of those are going to live long enough to be able to lay eggs on hawthorn. And hawthorn maggot flies, by the time they're sexually mature, there's almost no apples they could lay their eggs on. So the upshot is that the apple maggot flies are isolated from the hawthorn maggot flies, not by a boundary in space, not by a barrier in space, but by a barrier in time. The apple maggot flies are adapted to come out earlier in sync with the time that the apples are ripe. And that means that they rarely interbreed with the hawthorn flies, which are adapted to emerge at a time when hawthorn fruits are ripe. So it's not a space barrier, it's a time barrier. It sounds kind of Doctor Who. It's a time barrier. So what we find when we look is that the hybridization rates between races is very low, about 6%. You'd probably expect that hybrids would be selected against because hybrids might come out at a time when they're not in sync with either apple or hawthorn. We know the allele frequencies are different at all loci studied, which means that they're not interbreeding much. There's low gene flow between them. And apple flies, as we found in the lab, actually do better if they're raised on hawthorn. Hawthorn is better quality food for them. Apples, however, provide more food, and because apples are bigger, apple maggot fly maggots can burrow more deeply out of range of egg-laying parasite wasps. And because they're larger, there's less competition. So apple flies still do better if you raise them on hawthorns, but apples have certain advantages as a home and a food source too. The punchline, by the way, is that in northern Michigan, there weren't any apples grown until maybe 200 years ago. Apples were introduced from Europe. They were brought over here by the early settlers, by the early European settlers. So up until about 200 years ago, maggot flies were all living mostly on hawthorn. This is a couple of hawthorns in the background, by the way, because that's all there was. And only in the past 200 years has there been anything other than hawthorn available. And some of those apple maggot flies have evidently undergone change and made the jump to specializing in apples. Now they mature in sync with apple life cycle, not hawthorn, and they interbreed very rarely with the hawthorn maggot fly. And this must have happened in the past 200 years. This is what we call incipient speciation. It's speciation caught in the act. We have these two separate populations that may not be separate species yet, but they're different enough. We can distinguish them as different varieties They've separated without there being a physical barrier. There's nothing stopping hawthorn maggot flies from flying to meet the apple maggot flies. There's no physical block. Um, what's happening is sympatric speciation without a geographical barrier, and we've caught it in about midway. We have two populations that are not quite separate species, but look like they're getting that way. So speciation can happen sympatrically if populations become ecologically isolated, adapting to different habitats. This may explain the origin of other wild species. That's a blueberry maggot fly at the left. Those might have arisen in a similar way. Another cases that have been looked at since the 90s, sticklebacks again. You saw sticklebacks earlier when I was talking about sexual selection. These are the ones where males that have lots of red on their bellies have offspring that are resistant to parasite infection. So we've talked about them. Sticklebacks in British Columbia, sticklebacks can live in salt or fresh water. They're pretty tolerant that way. And 12,000 years ago, there was ice covering almost all of Canada, including British Columbia on the western coast between Washington State and Alaska. 12,000 years ago, that was all ice. The ice has retreated, it's melted. There are now lakes in British Columbia, and many of the lakes have been colonized by stickleback fish moving in from the ocean, where there have always been sticklebacks. So we now have a lot of stickleback populations that have colonized these lakes, and any changes that you see in these sticklebacks have only come about in the past 10,000 years or so. Here's the odd thing. Most of the lakes have two separate populations of stickleback. There'll be a population that lives up in the water column close to the surface. That's called a limnetic. And there'll also be a benthic stickleback living down on the bottom. And limnetics and benthics look different from each other. Up there at the top, you've got a male limnetic stickleback. You can see he's got overall kind of a lighter build, differently shaped mouth, more spines, more slender appearance, ventral spines right down here. The male benthic stickleback is thicker bodied, less spiny, right? Fewer spines on the back. And again, different shape of the head, different shape of the mouth, probably having to do with different diets, or at least that would be my guess. So each of these lakes has a limnetic population and a benthic population, and the two look different from each other. So here's the thing. We know this must have come about within the last 12,000 years or so, probably a bit less than that. We know there's no physical barrier separating the benthic and the limnetic sticklebacks. Any one of them could swim to encounter the others. There's nothing stopping them from mingling. And we do know that benthic and limnetic sticklebacks can interbreed. The hybrids are fertile. So biologically, we still call these the same species. The difference is that courtship behaviors are so different between the two that males of one population don't breed with females of the other. They attack females of the other. Male sticklebacks can be quite aggressive. So in the wild, they rarely interbreed because males of one attack females of the other instead of breeding with them. The strains, as you've seen, look different. They feed on different foods. They select different nesting sites. Hybridization in the wild is very rare. So even though they can hybridize, they rarely do. And there's some evidence for selection against the hybrids. It's been claimed that this would be, remember, speciation by reinforcement is what you get when two populations are driven to become more and more different from each other by selection against hybrids. So again, we've got two populations that are not quite really separate species, but are starting to look like they are. We know that it's happened only in the past 12,000 years. And we know that it's led to physical differences between the two. And we know there's no barrier between the benthic and the limnetic fish. There's nothing stopping members of one population from swimming to join members of the other. They just don't. So that's another case of sympatric speciation. Most recently, there's been a case of speciation observed in Baltic Sea populations of European flounder. That's a European flounder, rather grotesque fish, which as it develops, it starts out as an ordinary fish. And then as it develops, its skull twists so that both eyes come around to the right side of the head. And the fish spends its life lying on its left side with both eyes moved over to the right and the mouth kind of twisted. So yeah, kind of a oddball fish, but well adapted for lying on the bottom. And I might add, quite tasty, I'm told. Here's the Baltic. This is in Northern Europe. Denmark is over here. There's Norway, Sweden. This passageway right here that I've just kind of highlighted, the strait leading you into the Baltic between Denmark to the south and Norway and Sweden to the north, that narrows is actually called the Kattegat. If you watch the TV show Vikings, you might know that Ragnar was the ruler of the realm of Kattegat. The real Kattegat is part of the ocean, and it's this narrow passage that connects the Baltic Sea with the North Sea, which is off screen to the left. So anyway, there's Ragnar right there. So the Baltic does have a connection to the rest of the ocean, but it's kind of narrow. There's not huge amounts of water being exchanged between the Baltic and the North Sea, and a lot of rivers flow into the Baltic. And because of many rivers flowing into it, the average salinity of the ocean is 35 parts per thousand. Over here by Denmark, it's 20 parts per thousand. That's brackish, but pretty salty. Up here at the top of the Gulf of Bothnia, with a lot of rivers that aren't marked on this map, but there's a bunch of rivers running into, here, I'll draw some in, with a bunch of rivers flowing into the Baltic. I'm kind of making these up, but this is where, rivers flow. Oh, there's the Neva River. St. Petersburg is right there. There's a huge amount of fresh water getting pumped into the Baltic. And up here at the top of the Gulf of Bothnia, or over here by St. Petersburg, the water's almost fresh, right? Average ocean salinity is 35 parts per thousand. We're down to 2 parts per thousand up here in these light blue areas. So we've got a gradient between very fresh water and still pretty salty water over here by the Kattegat. Here's the thing about flounder. These are flounder eggs. Down here, you can see that this embryo right here still looks like a pretty ordinary fish. It hasn't started having one eye, the left eye twist over so it's on the right side of the skull. Flounder eggs are too dense to float in fresh water, but they do float in salt water. So flounder living in salt water produce eggs that float in the open sea, and we call this pelagic spawning. But flounder in the more freshwater parts of the Baltic lay their eggs on the bottom because the eggs don't float. That's called demersal spawning. So flounders lay their eggs in, flounder eggs either float or don't float, depending on whether they're in saltier water or fresher water. The upshot is that flounders that live in More salty areas, pelagic flounders, almost never breed with flounders that spawn on the bottom, the demersal flounders. So flounders that are demersal spawn on the bottom and lay their eggs on the bottom. Pelagic flounders may spawn up in the water column and release their eggs up in the water column. So they're breeding in different areas. Pelagic and demersal flounders almost never interbreed in the wild. What you're looking at here is you can take the frequencies of multiple alleles in multiple populations and boil those down to a couple of numbers that express most of the uh, variance in the data. And these are called the principal components. And so what these red dots are showing is you have all of these demersal populations clustered over here, and all of these pelagic populations clustered over here. And then you get the same thing here and there. What that means is that if you look at the frequencies of a whole lot of alleles, they're very different between the demersal flounders and the pelagic spawning flounders, meaning that there's very little gene flow between the two. There's virtually none. There's evidence for selection on demersal flounder genes that are connected with adaptation to low salinity. Evidently, the demersal flounders, they're the ones living in the fresher parts of the Baltic, and selection has acted to enable them to tolerate fresher water. And the demersal flounders, this is one of them, have recently been named Platyctes solimdali. They're considered different enough that they're now called a new species. And the Baltic only opened up to the world ocean in the wake of glaciers melting about 8,500 years ago. So solimdali must be younger than 8,500 years. That's well within the time that humans have been hanging out on the Baltic. Yeah, there were plenty of Stone Age hunter-gatherers and fisher folk living on the Baltic shore 8,500 years ago. And this is one of the fastest cases of speciation ever recorded in a vertebrate. And it looks like it was sympatric. There's no physical barrier blocking the demersal flounders from mingling with the pelagic flounders. So you can get speciation just by selection like that. And again, it may seem as if it's impossible to come up with a working definition of species, but I can't stress this enough. This is what we'd expect to find if evolution's happening. We should always have cases of species that are mostly different, but not completely, or kind of different, but mostly not, but a little bit, but we ought to have ambiguity in our species definitions. And Ernst Mayr, who started all of this by coming up with the allopatric speciation model, in his later years became a noted historian of science and pointed out that this is a shift from typological to population thinking. And it's an important event in the development of the way evolutionary biologists think. So start with Carl Linnaeus right here. When you saw that video back in the very first week, you might remember him. He was a Swedish physician. He ended up in the Swedish royal family as their personal physician, taught medicine at the University of Uppsala, which involved teaching botany because most medicines were derived from plants. So you had to know your botany if you did not want to prescribe the wrong plant and end up killing your patients. It helped matters that Linnaeus had an ego that you could not fit through a revolving door. The guy, self-esteem, low self-esteem was not a problem Linnaeus had. And he saw himself as being like the Blues Brothers on a mission from God. This is Linnaeus writing about himself in the third person, which Dr. Wagner thinks is creepy. God has allowed him to see more of his created work than any mortal before him. God has endowed him with the greatest insight into natural knowledge, greater than any has ever gained. Linnaeus is talking about himself here. Clearly he thinks that he has got, he's on a special mission. He's the one who's going to figure out how to classify all of life. And by doing that, he'll be able to get inside God's head, understand God's plan for his creation. To do this, he went back to Aristotle. There's Aristotle, the ancient Greek philosopher, who'd said if you have a bunch of things, the first thing to do is figure out what makes all of them alike, and we call that their genus. And then you figure out what makes each member of that set of things different from all the others, and we call that their species. So Aristotle belongs to the genus humanity, or man, because all men have 2 eyes and a nose and 2 ears and so on. He belongs to the species Aristotle because he has features about him that are different from everybody else. He's got a bald spot. He's got a big nose. He's got a beard. He goes around bothering people and philosophizing. He studied with Socrates. He supposedly once served as tutor to Alexander the Great before Alexander the Great decided that conquering the world was more fun than studying it. So there's all these things that make Aristotle different, and that is his species. But there's all the features that he has in common with other humans, and that is his genus. Linnaeus, of course, applied this to biological classification with a somewhat more restricted meaning. A species was, the basic type of living thing. Similar species could be placed into common genera, and then genera got placed into orders. Family came later. Orders got placed into classes. Classes got placed into kingdoms. The phylum was invented later. But you know that hierarchical classification system from other courses, all right? I hope you do, right? Kingdom, phylum, class, order, family, genus, species. King Paul came over from Germany swiftly. Kinky perverts come out for good sex. However you choose to remember it, you've all heard of that Linnaean hierarchical classification. Well, it started with Linnaeus's book Systema Naturae. The 10th edition of that book came out in 1758, and it was the first to use binomial names throughout. That's the starting point for animal taxonomy. Any older names aren't considered valid. The oldest Latin names that we still consider to be taxonomically valid come from that 10th edition of Systema Naturae. This is a page from, I think, the 7th edition, an earlier one. You can see over here it says Ordo, that's order primates, and he describes the order dentes primores superiores quatuor paralleli mammae pectorales binae. Basically means 4 parallel upper incisors, nipples on the chest, and there's a pair of them. Teeth and nipples are very important to Linnaeus for classifying his mammals. And then here's the first genus, Homo, which he just describes as nosce te ipsum, know yourself, recognize that this is you we're talking about. And his 2 species are Homo diurnus, which he would later replace with Homo sapiens, and Homo nocturnus, nocturnal man, which he identified with the orangutan, although the orangutan's now been put in a separate genus, genus Pongo. So there's his species. They go in the same genus, Homo, and they're placed, he didn't use families, so they're placed in the order primates. And these, of course, go into a class and then ultimately the kingdom Animalia. However, Linnaeus never doubted the biblical creation story. He did eventually get around, after studying his plants, he got around to the idea that new species might arise by hybridization. But for him, speciation by hybridization, it produced new combinations of what was already there, but it couldn't create anything radically new. Just like you can shuffle a deck of cards and deal out new combinations of cards, but all of the shuffling and all of the dealing, do it as long as you want to, you're never going to get an 11 of hearts, or a 12 of clubs, or a 6.5 of diamonds, or a plumber of spades. There's a king, a queen, and a jack, but shouldn't they have like a plumber of spades, or an accountant of spades, or a uh, short order cook of spades, or something like that? You're never going to get anything that's not already in the deck. And in the same way, maybe you could get a few new species by hybridizing, but that was only shuffling diversity that God had created in the Garden of Eden, just like it says in the book of Genesis. Linnaeus incidentally claimed that the Garden of Eden must have been a mountain, because that's the only way that you could have, Arctic species and tropical species living in the same area. The only way you're going to get that is if they're living on a very steep mountain with Arctic habitat on top and jungles down at the bottom. So he never doubted that each species, except for a few hybrids, must have been created separately by God in the Garden of Eden. He's not really an evolutionist. And since Linnaeus wanted to understand the plan of creation, he defined each species by some kind of intrinsic nature that God had defined. The condition for order in nature was the invariability of species. Species couldn't really vary. I mean, they can vary a little bit, like all humans look different, look the same. Or, okay, backwards. They can vary a little bit. All humans look a little different. All cats look a little different. But that variation is strictly limited. Species can't vary outside of pre-established limits. Nature can't color outside the lines. And every species is defined by these limits that God had created, these limits that define the species' essence. There's some sort of intrinsic nature. Cats may have different builds and different fur colors, but that variation only gets in the way. What you have to understand is that they are all within the limits of this thing that God made at the beginning called Felis Catus. They're all expressions of this essence of what it is to be a cat. And if you can understand what it is to be a cat, if you can understand the true essence of catitude, you'll understand just a bit more than you did about what God was doing when he made cats, assuming God made cats. I have questions. Species vary, but variation's ultimately not really relevant. This happens to be a Cuban land snail. They're all classified as one species, Polymyta picta, even though you can see the shells vary tremendously in color, in number of stripes and bands, even in proportions somewhat. All of this variation doesn't matter. What matters is the unchanging, divinely inspired nature of each species, its essence. This has still remained in biology, because whenever you name a new species, you have to find a specimen of it that shows, as best as possible, the features of that species that you think are most important. And you call that the type specimen. and the type specimen is kept very carefully, and it is supposed to be deposited in an established museum. This one over here, this sheet says Herb Hort Q. That is Herbarium Hortensis Quensi, the herbarium of the Royal Botanical Gardens at Kew Gardens in the south of London. So this type is kept on a pressed sheet of paper. It's kept very carefully. Types are usually not put on display. In fact, several of the museums at the University of California didn't have very much in the way of public display. Maybe just a couple of cases, maybe not even that. They didn't do much work for the public at all. They're not what you might think of as a museum, but they kept large collections of organisms. And in particular, they kept many type specimens. And the type is forever the standard reference for defining a species. If there's ever any debate as to this species that has been named, somebody's going to go back to the type and use this as the standard of reference to compare other specimens with. So if there's any doubt as to what did the guy mean by such and such species, it goes back to the type. If you collect a similar plant and you say, I think this is a different species from this one, you might actually have to travel to that museum and study the type directly. So the type is forever the standard reference for defining what that species is. You also get this in the thought of modern creationists. The Creation Research Society, to be a member, you have to sign on to their statement of belief, which is that all basic types of living things were made by direct creative acts of God through creation week. Whatever changes have occurred since creation week have accomplished only changes within the original created kinds. This is typological thinking. It's looking at diversity as reducible to a fixed and finite set of types of things. Variation among individuals may be real, but it doesn't go beyond the borders of these fixed types. All living things are expressions of a limited number of kind of ideals or essences. To an evolutionary biologist, however, variation is critical. Species do change. Variability is what makes that possible. Darwinian evolution would fail to work if organisms didn't vary, but in fact they do. This happens to be a collection of a moth from southern Arizona called Semeiopus ella. These have been mostly collected from the same area, Big Bend National Park, and you can see they vary. There are some that are deeper colored, some that are lighter colored, some that have more spots, and some that have fewer spots. And this is just one species in one area. Individuals vary. Variation is inherited. Variation is what makes natural selection possible. And sometimes that variability just slops over the edges of the nice, neat definitions that you'd like to draw. Darwin went so far as to suggest that species should not be considered to objectively exist at all. Species are, well, a little bit like colors. You know, if you think about it, we have customary names for colors like, you know, yellow, green, blue, and things like that. But if you actually make a color spectrum, you can, you can find shades that are in between. You know, you can find greens that kind of shade off into blue, and there's colors that you might not really be able to pin down as either one. You know, how do you classify a turquoise or aquamarine or shades like that? Um, red and red and yellow are considered separate colors, but they kind of fade into each other. We usually talk about red, orange, and yellow, but there's all kinds of in-betweens there. There's, vermilion and just, reddish orange. There's really a complete spectrum. And we give names to different parts of the color spectrum: red, orange, yellow, green, blue, indigo, violet, Roy G. Biv. And other cultures give different names and divide up that spectrum in different ways. But ultimately, it's just a matter of convenience where we decide to split up that spectrum. You know, red is not real. What's real is a bunch of wavelengths of light that stimulate the photoreceptors in our eyes. And we've all kind of agreed to call, wavelengths of light stimulating your retina at certain frequencies red. But there's nothing, you know, there's no natural definition of red because colors exist on a spectrum. Darwin suggested that species do too. As he put it, the only distinction between species and varieties is that varieties are known or believed to be connected by intermediate gradations, whereas species were formerly thus connected. We shall have to treat species in the same manner as those naturalists treat genera who admit that genera are merely artificial combinations made for convenience. This may not be a cheering prospect, but we shall at least be freed from the vain search for the undiscovered and undiscoverable essence of the term species. So Darwin just says species don't have an essence. Life varies, individuals vary, populations vary, and it may be convenient to cut out a chunk of that variation and call it a species, just like it's convenient to take a subset of all of the wavelengths that stimulate your retina and call it red, but there's no objective place to draw the line, right? Just like red shades off into orange, species shade into each other, and species aren't, according to Darwin, objectively real objects at all. And Ernst Mayr has emphasized that this is a shift from typological thinking to population thinking. Population thinking, variation is everything. It's not something that gets in the way. It's the beating heart of what you're trying to do. Living things vary, populations vary, and instead of regarding variation as a nuisance, it's at the very heart of your research. You try to understand why things vary, how the variation is shaped by selection, among other things. Thinking of species as sets of changing and varying and shifting and blending populations instead of being this fixed number of essences. The last dead white dude I want to introduce you to is Alfred Kinsey. Kinsey was an entomologist at Indiana University, and he was a specialist in cynipids. This is a cynipid. It's a wasp, doesn't sting, harmless, can't hurt you. And like the sumac gall aphids, cynipids lay their eggs in plant tissues. Specifically, cynipids almost exclusively lay their eggs in oaks. The egg, once it's been laid in an oak leaf or bud or twig, stimulates the plant to start growing more tissues around the egg, and then the egg hatches into larvae that eat the plant tissues. Any oak tree in summer, look long enough and you'll find cynipid galls on the twigs or the leaves. The largest type you do sometimes see, usually among fallen leaves in the nature reserve. They're called oak apples. These round swellings on oak leaves that can get up to the size of a golf ball. When you see them on fallen leaves, they're usually empty and hollow and kind of papery. But yeah, that's cynipid galls. Here's the thing, there's lots of cynipid species, but most of them had been defined on the basis of only a couple of specimens. Every species was defined, as he put it, by a half dozen specimens pinned in their cabinets. So an entomologist goes out, collects a handful of gall wasps, decides that a couple of them look different from everything else, names it as a new species. And as Kinsey put it, these are the systematists responsible for the definition of systematic entomology as the science of transferring pins from one box to another. What Kinsey, his students, and coworkers did was drive all over the United States every summer from oak grove to oak grove and field to field collecting wasps. And they ended up collecting over 71,000. So instead of basing cynipid species on just a couple of specimens that look funny, what he tried to do was capture the entire range of variation among the cynipids. Are there really natural places where you can divide up the cynipids, or does every species blend into others? So what he concluded was that even though species could change, very often you still could define species pretty well. There were some natural clumpings and clusterings of his wasps that seemed to correspond to species, and he compared his wasps to streams. You know, a stream is constantly flowing. It may change its course. Some streams flow into other streams. Streams might vary in their speed or their currents, or whirlpools and eddies or things like that. Sometimes your stream might fly through a really marshy area and it might be really hard to tell exactly where it went. But still, it was useful to define the course of a stream or a river. And species were like that as well. Species can blend into each other, but very often you can find these kind of natural clusterings that correspond to species, even if it's not always possible to define them perfectly. Incidentally, the reason I brought Kinsey up is Kinsey's actually famous for a different type of research, although I have read his studies in the gall wasp genus Cynips. Very good book if you're interested in galls. Kinsey was a professor at Indiana U, and a lot of universities used to offer courses in, they'd call it family life or marriage and the family. What it basically was sex education. They didn't call it that back then. But, I guess by college age, you were deemed to be mature enough to be able to withstand the shock of knowing how you make babies. Okay. And be taught how to make babies properly. Right. So Kinsey, like a lot of biologists, just about every academic biologist has to teach lower division courses not in the field of specialty. I teach biology 1400. Some of us teach, 1440, or, anatomy and physiology for the health science majors, things like that. Kinsey had to teach this course in what was basically human sexuality. And he was a conscientious man. He tried to do a good job. He looked up what was known about human sexual behavior. And this was back in the day when books on human sexual behavior used to be written in Latin so that only the most educated people could even understand them. So that, uneducated people or, women would not be able to understand what was even being talked about. And that's not that uncommon. Even if the book was mostly in English, the really zippy bits tended to get placed in Latin. And Kinsey reviewed all of this and found that there was literature, there were written descriptions of things that were considered abnormal sexual behavior. You could find case reports about, people who could only get off by wearing rubber raincoats and smearing whipped cream on their noses or whatever it is. But people had tried to understand what was considered pathological, what was considered diseased, but nobody had done any studies of what ordinary people actually did. There's, studies were available on, Hamish the Scottish sex maniac, but nobody had bothered to look at what ordinary, regular folks actually did. And Kinsey applied the same approach to sex that he had to gall wasps. He'd collected over 71,000 gall wasps to try to determine, okay, what is normal for this species and what isn't? What's the total range of variation in the phenotype of gall wasps? Well, he sent out thousands of questionnaires to try to determine what are the ranges of variation in human sexuality. And he published his results in two books in the 1950s called Sexual Behavior in the Human Male and Sexual Behavior in the Human Female. Sexual behavior in the human female got him personally denounced in Congress. The state of Indiana even threatened to stop funding the university unless they fired him, because sexual behavior in the human female did things like reveal that premarital sex was a hell of a lot more common than anybody was comfortable admitting. This is the 50s. This is still the time in which, good girls keep themselves pure for marriage. The truth is, rates of premarital sex actually have not changed all that much between your grandma's generation and yours. People didn't talk about it back then, but it was a lot more common among nice girls than anybody was prepared to believe in the 50s. And Kinsey was seen as, having, smeared the purity of American womanhood. Kinsey found that the answer to this question depends on how it's worded and on how it's defined. Kinsey was the one that found that 1 in 10 human men have had homosexual encounters. Not all of them would necessarily call themselves gay. And again, the number that you get depends on exactly how you phrase the question. But homosexuality was a lot more common than people were comfortable thinking of. Sexual contact between humans and animals was surprisingly common, especially on farms. Kinsey, not a very humorous man, but he did claim that according to his results, 85% of the population of Indiana should be in prison, and the other 15% were anemic. In other words, they didn't have the physical vigor to do what they wanted to do. Kinsey's work has been revised a great deal. He did stay there. He did end up founding a major institute for the study of human sexuality. The Kinsey Institute is still there at the University of Indiana. They've made research into human sexuality a respected field. There's any number of books and courses that you can take that basically cover what the full range of sexual behavior in humans. It's a much wider range than people wanted to admit in the 50s, but it's something we understand a lot better now. And it ultimately comes from the same kind of inspiration that Kinsey was using to understand his gall wasps. The idea that instead of restricting what you're studying to a few neatly defined types, you've got to try to encompass the entire spectrum of variation. Variation's at the heart of the matter. Variation's the most important thing. And you're not going to understand life, whether it's gall wasps or people getting their freak on, you're not going to truly understand it if you don't have an appreciation for the full range of diversity, because it's in that range that understanding ultimately lies. Oofda. Okay, that got a little ranty at the end, but I'm going to go ahead and that's the end of the show. So I'm going to stop recording. Uno, dos, three.